Hi everyone, welcome to this week's Meet a Scientist live stream. We're coming to you live from the Youth BIOlab at the Albertson Research Center at St. Boniface Hospital in Winnipeg, Manitoba. We're on Treaty 1 territory, the land of the Anishinaabe, Cree, Oji-Cree, Dakota, and Dene peoples, and the homeland of the Métis Nation. We're also very privileged to be safe and healthy right now, and we hope that you are doing as well as can be at this time too. In this weekly live stream, we get to have conversations with people from around the research center so that we can learn a little more about the work that goes on here, but more importantly, learn a little more about the people who do the work here and a little bit about why they love the work that they do. I'll be keeping an eye on the chat as well, so if you have any questions, you can join us there too. Today, we are joined by Don Davies. He's a postdoctoral fellow in the Laboratory of Synaptic Plasticity and Memory Dysfunction, which is part of our Division of Neurodegenerative Disorders. Welcome, Don, and thank you so much for joining me here today.
Thanks for having me.
Yeah, it's great to see you. So Don is relatively new to the Research Center. He is a postdoctoral fellow. So Don, can you tell us a little bit about what that actually is?
Sure, yeah. So a postdoctoral fellow has completed a PhD. So I've got my undergrad, master's, PhD, and then when there's, searching for a professorship, there's not always one available. So I'm a postdoc, which is basically like an intern to hold me over for, 2 years, 3 years, however long it takes. To find a professorship or a job in industry.
Okay, so your goal is to teach?
It is, yeah.
At university, all right. So as a postdoctoral fellow then, so you're working in this lab sort of as an intern, you said, so how do you fit into the lab work, or what do you do while you're working in the lab?
So I lead various experiments in the lab and have students under me that produce data. I also produce data, writing grants, writing manuscripts for scientific publications. So a variety of different roles I play.
Okay. And so I know that you moved here during the summer and you've kind of been bouncing around. Can you tell us a little bit about what your journey has been to get to even this point and sort of where you hope to go from here? You said professorship, but if your area of study is brains, how did that kind of take you all over the place and how did you end up here?
Yeah, so I originally grew up in Winnipeg and got my undergrad in University of Winnipeg. Got a psychology degree that wasn't sciencey. It was only looking at behavior. And it wasn't until I got into my master's at the University of Saskatchewan in Saskatoon that I really began looking at neuroscience and the brain function. And then from there, after my master's and PhD, I did my first postdoc at the University of Western Ontario in London, where I did that for about 2 years. And then finally back to Winnipeg to do another postdoc. So this is my second postdoc.
Okay. Do you learn different areas of neuroscience in all of your postdocs, or do you have a specialty that is like your area of expertise and you bring that with you to wherever you go?
Yeah, it's always been learning and memory has been the central theme of it, but there's always another layer that I'm learning through each postdoc. Like my last postdoc was looking at gut-brain axis and how the gut communicates with the brain and alters memory and other behavior. So, and then this postdoc is looking at like novel compounds used for Alzheimer's disease. So it all has to do with learning and memory, but there's different side components that I'm learning.
When we talk about disease with students, we often refer to the approach to medical research as like a big puzzle. So there, even if the problem is memory and age-related memory loss or Alzheimer's, there are lots of different pieces to that puzzle. So you studied the foods piece, you've studied behavioral pieces when you're studying psychology, and now you're looking at the actual brain and how it works. Have you always been interested in like memory related problems, or what made you feel like pursuing a science career in the first place?
Yeah, I know during high school, I was always interested in psychology and behavior in general. So I was always thinking when I was in high school that I was going to become a psychologist. And
Mm,
yeah, then as I went through undergrad, I realized I was more interested in how the brain functioned and how memory and the brain connected, or the molecular mechanisms of memory.
mhm. Super complicated. So can you tell us a little bit more specifically then about what your lab work is doing? You kind of study, you've sent me a few things and said, you're studying the lab is studying synaptic plasticity and memory dysfunction. So for people, sometimes people already have like, oh yeah, the brain is plastic, it changes or whatever. But can you explain a little bit about what synaptic plasticity is and how it's affected in aging or in diseases like Alzheimer's?
Yeah, sure. So, synaptic plasticity is just either a strengthening or a decrease in the connection between neurons. Um, since memory is stored at the synapse, and a synapse is just a gap between a presynaptic neuron and a postsynaptic neuron. And there's various neurotransmitters and signaling modulators such as calcium that go from the presynaptic neuron to the postsynaptic neuron.
Hmm.
And when we see excitatory neurotransmitters and signaling molecules moving across the synapse, we see an increase of firing and increase in synaptic strength, which correlates to memory.
Hmm. So is it like is it like musculature? Like if you use those synapses more, they get stronger or
Yes, definitely. If you use them more, they tend to get stronger. So yeah, some of the analogies is like driving on a road and making ruts, and like how um, you're just making it stronger and stronger over time. So the more you use those connections, the stronger they become over time.
Hmm. I made a little doodle that I'll share with people just in case people aren't familiar with nerve cells or with synapses when Don's talking about his research. Here's a little nerve cell diagram. Not all nerve cells look like this. There's different types of nerve cells, but the basic structure is, there's a cell body and there are these branches up top called dendrites. So those would be receiving the signal. And at the beginning of each little tiny branch up top, you would have, another, what did you call it? The receiving end? What's the term for
Oh,
that?
the
Postsynaptic?
postsynaptic neuron, so it would
Yeah.
be an axon.
So down here, so this is the axon. That's your presynaptic neuron. And then there's a little gap, right? So you're studying sort of how able these synapses or the connection between the two neurons are able to give those messages to the next cells. But you said, so the memory is stored in the synaptic terminal. Can you explain what that means a little bit more? Because that's something new that I haven't necessarily heard before. I'm going to stop sharing so it's just us talking again.
Yeah, so I guess maybe many people think that synapses are this fixed structure, which isn't really true. Every time you learn, there's usually new synapses being made. So the
Hmm.
more you learn, the more synapses are being made. It's not necessarily new neurons, although that does happen in certain areas of the brain, such as the hippocampus, new neurons being created. But the literature is showing that that memory is being located at the synapse, at the junction between the neurons as a molecular correlate to memory.
So, wow, that's fascinating because we often hear, your brain cells aren't growing quickly. We talk about how important it is to protect them all the time. So I think a lot of people do get that idea that, you have X number of brain cells and they just sit there stationary, like sending their little signals when they need to, but they're actually growing new connections, maybe similar numbers overall, but they're branching out more. That's the picture that we can draw from this.
Yeah, exactly. Their neurons are constantly creating new synapses and new connections among other neurons.
Okay. And so you sent me a little bit of information earlier looking at how that plasticity is affected by old age and Alzheimer's disease. Can you explain some of those changes that you see in the synapses as people age?
Yeah, so there's various, I guess, classic hallmarks of Alzheimer's, such as plaques being deposited in the brain, A beta plaques, and then neurofibrillary tangles, which is almost you see this corkscrewing of a neuron retreating so that the synapse aren't really functional anymore. The neurons aren't functional because they don't have their partner neurons. So there's that, and then there's this level of inflammation that we see before we see the plaques and tangles in the brain. So one of the possible potential therapeutic avenues would be to lower the inflammation
Can I jump in and
before.
ask a question?
Mhm.
So you said you see that inflammation before people actually see the plaques and tangles. When does that begin happening in people? Because even measuring the plaques and tangles, from what I understand, is very, very tricky. So diagnosing Alzheimer's in someone almost depends on taking a piece of brain out and looking at it to find those physical plaques and tangles. And uh, presumably those plaques and tangles start happening long before the extreme symptoms of memory loss show up. So like, I'm 35, should I start worrying about the levels of inflammation in my brain now to avoid these problems in the future?
Um, I don't think it's necessarily, I think it's something that, everyone should be thinking of, like inflammation and how they can, um, I guess, minimize it given lifestyle factors such as diet, nutrition, stuff like that. But yeah, I don't think we're really at the point now where we can say, I'm 35 and I really got to be thinking about how I'm going to prevent Alzheimer's when I'm, 50, 60, whatever it is.
Mm.
I don't think the science is really there yet, although it has been shown that there is this low level of inflammation that is correlated
Mm.
with Alzheimer's disease later in life.
I would like to know how to prevent Alzheimer's in myself. I've had a few concussions, so anything that you hear, let me know. So what are some of the things that you're looking at then to help those neurons? Actually, I think I interrupted you while you were talking about it. Can you explain again uh, how the inflammation affects the plasticity?
Yeah, so when um, there's too much inflammation or an abnormal amount of inflammation in the brain, you can get impaired synaptic plasticity, which would lead to impaired memory. So perhaps if we can decrease the inflammation in the brain, it would prevent the plaques and tangles from occurring, and we would hopefully prevent Alzheimer's from occurring.
Hmm. When you say you can prevent the decrease in synaptic plasticity. Like, is the decrease because there are fewer connections, like fewer actual synapses, or do some of them just not convey messages as well? What are
It seems to be that they're just not conveying the messages well. When there's a heightened level of inflammation, there is disruption in synaptic plasticity. So even though they're making the connections, they're not strengthening. So even if you're doing the same behavior, like trying to learn a specific task to increase synaptic plasticity, it's if you have this inflammation, it's most likely not going to be that effective.
Well, let's get to some of the solutions then. You said you've been working on some novel or new compounds that we think might help maintain synaptic plasticity with aging. What are some of the things that you're working on now, and where did those things come from? Like, what work are you growing on?
Yeah, so we're currently in the process of getting an NRF2 activator, and then NRF2 is a transcription factor which regulates, turns on a gene for antioxidants, various genes related to antioxidants. And so the theory is, or the hope is, that we can use this compound to decrease inflammation and improve memory on various memory tasks and synaptic plasticity.
So, okay, I'm going to backtrack there because you just said a lot of things. So we hear about antioxidants in food, all like in ways, antioxidants are good for you. They're in lots of the like really bright colored fruits and vegetables, like some of them make things, the red cabbage purple and all these things. And they've been known to maybe help prevent cancer. Some of them are good for the heart. But our bodies are actually making antioxidants as well.
Yeah, exactly. So you can turn on genes to make antioxidants depending on what foods you eat. So I know sulforaphane in broccoli is one example that has come up from time to time in the media as being a potent NRF2 activator. And people will eat broccoli with the hopes of decreasing inflammation.
Interesting. So transcription factors are something that, is just like a little off from what students are learning usually in their grade 12 biology. When they study DNA, we often learn like transcription and translation. So that's like the reading of DNA, sending it with mRNA to the ribosomes, and then the ribosomes make proteins. But what is a transcription factor? Like, what is it changing in the cells?
So it binds to certain regions of DNA to turn them on or off, like a light switch almost,
Yeah.
so that they can bind and either express certain genes or repress certain genes. And then in our case, with the NRF2 activator, we're interested in expressing a gene. So we're interested in having NRF2 bind to a specific region of DNA and turn that gene on to create messenger RNA for an antioxidant.
So NRF2 is like a little factory worker that's going and turning the switches on high to produce antioxidants, reading those
Exactly.
specific genes for antioxidants. What are some of the other things? So you said sometimes something in broccoli, sulforaphane, can help stimulate that little factory worker to go flip the switches. What are some other things that people can do if we hate broccoli, or you're working on a novel compound, a brand new synthetic compound maybe that that could do the same thing, why not do the natural choice? Or like how do you go from seeing this thing in broccoli and saying, oh, well, I need to make that better by making something in a lab? But like, what's going to be the difference with it?
Yeah, so I guess the thing in the lab, making it synthetic, gives you concentration, so you know the dose, and you
Mm,
would be able to know the frequency, whereas broccoli, um, people don't really, the majority of people probably don't eat, say a certain number of, a certain weight of broccoli every day and, get the dose correct every day. Whereas
mhm.
a synthetic compound could be prescribed in drug form and people could take it every day if they were at risk for Alzheimer's and other dementias.
Hmm. So, okay, so I would have to eat like a kilogram of broccoli a day, maybe, or more or something to get that amount of the beneficial compound in it. That might be really good for my gut bacteria too, but that seems like a lot. Um, so what are some of the techniques then that you use in your lab? Like, how do you know if this new compound is working or not? What are you actually measuring?
So we're measuring synaptic plasticity. So we use electrophysiology to look at brain waves and to see if the brain waves, it's called long-term potentiation, but it's just a change in synaptic strength so that we can measure if the NRF2 activator is increasing synaptic plasticity. And then we also use a variety of behavioral tests, one of them being the Morris Water Maze, which is a apparatus where it's basically a pool for a mouse, where the mouse is placed in a pool and can swim to find a platform, a hidden platform. And that's a measure of memory, how long it takes to find the platform. If it takes a very long time to find a platform, it has poor memory. If it quickly finds the platform, then it has good memory.
Hmm.
So there's, yeah, the memory tests and the synaptic plasticity that we can measure.
Hmm. So has your lab started testing any of these NRF2 activators in any
Not
models?
yet. We're hoping to get a shipment fairly soon. The the NRF2 activator that we're receiving is in New York, and it's
Mm.
with the border
Mm-hmm.
security has been, yeah, it's, and because it's a novel compound, um, it's having a little bit of a hard time getting across the border, but
Hmm.
yeah.
Has anyone just fed mice a bunch of broccoli to see how much they would have to eat to improve their memories?
I'm not sure about that. That's a good question. That'd be a good experiment though, to,
Yeah.
yeah, feed mice
Sometimes
broccoli.
we study these things in the actual foods. So you're saying, so research has definitely been affected by, COVID and the difficulty in getting things shipped, and you said it's a novel compound and whatnot. Um, you've also been a little bit affected by the COVID. So Don moved to back to Winnipeg to join this lab in the middle of the summer during the pandemic. And our research center, it's very empty looking behind me right now, because I have to be alone in my lab during our COVID precautions. But we're typically like a very social, like family type group. We have lots of festivities. What was it like for you to have to move and join a new lab during this time when everyone is socially and physically distancing?
Yeah, it was a different experience for sure than any of my other labs that I've joined in the past. Because in the past, you just go to a lab and it's very social.
Mhm.
Whereas this lab now, everything was done over Zoom or over phone calls, so
Mhm.
it was a bit of adapting period to get used to doing everything over Zoom and asking, questions over Zoom instead of actually being in person for the most part.
Mm-hmm. Are you doing a lot of your, like, is your research right now not as much benchtop research, but you're able to work remotely and kind of do some stuff at home?
Yeah, I do. I'd say it's about half and half now, where I do half my work at home and then half my work in the lab. And then even in the lab, we have to be socially distant and make sure that there's not too many people in the lab at the same time.
Mhm. For our viewers too, anytime we're not just alone in a big space, we have to be masked up as well. So we have a lot of safety precautions here, and we're lucky to access lots of PPE to make sure that we are safe and clean. And we have excellent cleaning staff, but it's still like everything is very affected by it. Hopefully by, this coming summer, we'll be able to actually have a bit more social activities and sharing of research and sharing of our, like people do their poster presentations and we get to see what everyone's doing. So it's been a bit of a weird time for everybody. But this is a really important part of the brain's research puzzle. And I know that this is probably a bit different now because you have joined the lab at such a weird time, but what are some of your favorite parts of doing benchtop research?
I really enjoy being able to discover stuff that I guess no one else has, seen before, and publish a novel result, and really building on the science that was done before me,
Mm-hmm.
and coming at it at a new angle, and just discovering something new, even if it is a small little mechanism to discover something new and publish it and have it have people talk about it. I think that's really cool.
Yeah, and that does seem, finding new compounds for Alzheimer's disease, memory is so complicated. And, like lots of Canadians experience dementia. It's a huge burden financially and socially on people. So that seems like very valuable work that your lab is doing as well. You kind of said, the idea of like imagining new things and seeing what's next. We have found by interviewing quite a few people here that they're quite creative. People typically picture, scientists as just being, like doing their lab work and whatever, but it is really a creative process, or imagining, what comes next. Do you see creativity as part of your work process, or do you have other creative endeavors that you think use those same parts of your brain?
Yeah, I think science is very creative. I know when I came on in the middle of a pandemic, and I was thinking of different projects to get started. I was thinking, I Googled like COVID-19 and the brain, and kind of to my surprise, there was actually a lot of articles
Hmm.
about it, scientific publications about COVID-19 and the brain. So
Hmm.
actually one of my side projects now is looking at COVID-19 and the brain, and where I'm working with a collaborator at the National Microbiology Lab.
Hmm.
And there's also this international collaboration with um, this PI in Philadelphia. So
Wow.
it's kind of been this thing that I didn't even plan to have happen. I when I joined the lab, I thought all I was going to do was Alzheimer's research, but
Hmm.
then the COVID-19 and the brain just kind of happened, just kind of thought of it, and almost out of nowhere. And now it's becoming this international team that
Yeah.
all collaborating.
That does seem to be an interesting effect of COVID in general now, that research institutions from all over the world are starting this like international sharing or doing co-projects. And that's really cool. Hopefully we don't see any incredibly negative effects for COVID on the brain, but it does seem to affect lots of things. What have you found in your research? We had a question from one of our people on our live chat. What does COVID actually do to the brain from the things that you've read so far?
So from the things I've read so far, it usually starts in the nasal cavity, and then it can work its way up through the olfactory nerve and go into the brain.
Really?
And yeah, and
That's
actually,
terrifying.
yeah. So it is kind of terrifying because then you see this increase of inflammation.
So
Um,
that is that why lots of people lose their sense of smell? The olfactory nerve, that's what leads to the scent processing area of the brain. Is that responsible for that?
Yeah, that would be responsible. I don't think the science is there to show like a causal relationship yet, but it would
Yeah.
make sense that if they can show, they have shown that it infects COVID-19 can infect the olfactory nerve and travel
Wow.
to the sensory areas of the brain, that
Hmm.
it would make sense that's why you lose your smell in some people with COVID-19.
Hmm. Does it lead to other more serious effects, or is that
Yeah,
understood at all yet?
I mean, it's still emerging, but from what I've read, there are some serious effects where it can, the way the olfactory nerve goes, it can go into the brain stem, and then there's respiratory centers there. So
Oh,
it can affect respiration in the brain.
wow.
Um, And then, yeah, maybe some doctors on the front line are thinking this is a lung problem or a heart problem, and that's
Yeah.
the reason for the poor respiration outcomes. But it might actually, in some patients, be the brain infecting the brain stem that controls the
Wow.
respiratory centers, and then COVID-19 impairing it.
Wow. Such a multifaceted problem. It seems like there's going to be a lot of research upcoming in all of the different effects of COVID on different parts of the body. I think that we're kind of roughly at the end of our interview time today for what we've told our viewers or anything. But I'll ask you one last question, and we sort of ask this to most people that are on our show. But what if you weren't working in science? Like, what else could you see yourself be doing?
I could see myself, maybe being in finance or something like that. I was
Oh,
actually thinking
interesting.
about getting an MBA, so maybe something more business related.
Okay. So you could learn how to sell the new drugs you're developing for lots and lots of money.
Yeah, exactly.
Yeah, we work with the for the university right now, so I think a lot of people don't understand that our labs don't sell drugs for money. But that sounds pretty good. Understanding money is also very important, not something that I'm very familiar with. But thank you very much for joining us today. And next week, we're going to have Rebecca Camargo with us from the Institute of Cardiovascular Sciences in the Molecular Cardiology Lab, and she's going to share a bit about her research on hearts. And you can make sure to subscribe to our YouTube channel for updates. Check us out at youthbiolab.ca or follow us at youthbiolab on Twitter or Instagram to keep up to date. Thank you for watching, everybody, and stay curious. All right, I think I